But Calvin Bridges in 1916 showed that the Y chromosome is not at all involved in sex determination. And this was a very, very important statement because till then it was considered that it is the Y chromosome in any organism, okay, except XO, XX, XO, Y is the determinant. Okay. So if Y is there, then it is going to be a male. If Y is absent, then it is going to be a female. That was the concept. But Kelvin Bridges totally ignored the presence of, not ignored, he mentioned clearly, I'm sorry, he did not ignore, he basically focused on Y chromosome, and he said in case of fruit flies or drosophila, Y is not involved in sex determination. So, how he did it? He looked into non-disjunction, and you will be surprised that Calvin Bridges was a student of whom? Do you know that?
TH Morgan.
Morgan.
Very nice. Yeah. He was a PhD student of Professor Morgan. So, Professor Morgan was working on sex linked inheritance. And I will tell you that this story when we will come to sex linked inheritance also. But since we are talking about Bridges, Bridges gave one of the most important theory, most, which is non-disjunction. Okay. And how he did it, that is really a motivational story for any student who is curious in nature. Whether that student is pursuing PhD or not has no meaning. But what was happening, Professor Morgan जब भी अपना काम कर रहे थे, तो अनएक्सपेक्टेडली उनको कुछ ऐसे जीनोटाइप मिल रहे थे फ्लाई में, कहीं कहीं वाइट फ्लाइज मिल जा रही थी, कहीं कहीं वाइट रेड नहीं मिलनी चाहिए थी, वहां रेड मिल जा रही थी. एंड व्हाट ही वाज डूइंग as a very senior professor also, he was only focusing on sex linked inheritance. Okay, so his hypothesis was that I have to look into the inheritance part and I have to prove that it is linked to the sex. And conveniently, जो कि mostly researchers do, जो भी unexpected flies आ रही थी was trashing them. But Bridges was observing it. Okay, and he started thinking about these flies which were very very unusual. देखो जेनेटिक क्रॉस क्या होता है, इट इज अ मैथमेटिक्स बेसिकली. जब भी आप 2 + 2 करोगे, नो मैटर व्हाट, इट हैज टू बी 4, राइट? Similarly, in genetic cross also, the genotype of the fly, whether you talk about F1 generation or F2 generation, it has to be as per your expectation. लेकिन जब नहीं आ रहा था, तो मॉर्गन उसको फेंक दे रहे थे. Calvin Bridges sat with those flies and he started looking into the genotype and phenotype of the flies. And then he came into this observation which is known as non-disjunction. और जितने syndromes आप लोग पढ़ते हो, that is understood only because of the non-disjunction. आप लोग पढ़ते हो ना, टर्नर सिंड्रोम पढ़ते हो, क्लाइन
Yes,
फिल्टर
ma'am.
पढ़ते हो, पॉलीएक्स पढ़ते हो, ये सब सब लाइट में तभी समझ में आया जब कैल्विन ब्रिजेस ने नॉन डिसजंक्शन की थ्योरी दी. नहीं तो किसी को समझ में नहीं आता कि ये एबनॉर्मल सेग्रीगेशन ऑफ क्रोमोसोम कैसे होता है. ओके, so although Morgan got Nobel Prize, and I feel really sorry for Calvin Bridges, his contribution is massive in the field of genetics and of course in human beings, because a syndrome, they are widely spread, है ना? लेकिन now we understand the basis behind it. ओके, सो वो चीजें हम लोग और पढ़ेंगे बाद में. लेकिन कमिंग बैक टू वाई क्रोमोसोम नाउ, तो क्या हुआ जब कैल्विन ब्रिजेस ने कुछ फीमेल फ्लाइज देखी, and these flies, later on he understood that it is happening because of non disjunction. He found that these females were XXY, okay, or they were XX, or they were XO, or even they were XY, okay. So he was kind of confused because whenever there is Y chromosome, the Y chromosome, then the it has to be a male. So now he started looking into this concept that along with various sex chromosomal combination with 3N autosomes in case of drosophila, uh, they were females. Or can you please tell me how many chromosomes are there in fruit flies in drosophila?
Ma'am, there are
Ma'am,
three
eight.
pair of
There are
autosomes
three
and
pairs
one
of
Okay,
pair of
one
autosomes
by
sex
as
one.
well.
chromosome.
Then
There
I
are
would
one.
like to hear the name. Yeah, one by
Ma'am,
one.
Sona.
Sona, yes, Sona, please.
Ma'am, like there are three pair of autosomes and one pair of sex chromosomes.
Very nice.
Means eight total, eight total.
Total eight. So total is eight, or you can also say four pairs. Okay, so it's our call. Both of them are going to have same number. So his observation was कि अगर Y होता भी है तो उसके बावजूद भी ये फीमेल होती है. And then he started thinking about it, and finally, of course, non-disjunction theory came into the light. But he also said that the presence of Y chromosome is not going to determine maleness. देखो, developmental biology के student बनोगे, I mean you are going to study that, you are a geneticist already, and you are going to study a lot of molecular mechanisms in different classes, okay. So you have to understand these words. When I'm saying maleness, then it is only related to the phenotype, okay. So he clearly said that Y chromosome is not going to determine maleness in Drosophila. Instead, each fly's sex is determined by the balance between genes on the autosome and genes on the X chromosome. Okay? ये आप लोगों ने बीएससी में भी पढ़ा है. उस दिन कई स्टूडेंट्स कह रहे थे जब हम ह्यूमन बीइंग की बात कर रहे थे तो आप ड्रोसोफिला के बारे में बता रहे थे. बट let us revise it once again. So, this type of sex determination is called genic balance system, and it was given by Bridges by his thorough observation. In this system, several genes are known to influence sexual development. The X chromosome is going to contain the genes with female producing effects, whereas autosomes contain genes with male producing effects. Okay, and therefore, fly sex is determined by X to A ratio. So, अब सब आप लोगों को पता है, but I am going to revise it so that it becomes clear to all of you uh, in our following classes, okay, when we develop a little bit of concepts. So how the calculation is being done? Sex determining ratio is equal to number of X chromosomes, okay, and it is divided by number of haploid sets of autosomes, okay. Now what is being considered? अब अलग अलग कंसेप्ट हैं इसमें. हमने कहा 3 पेयर्स ऑफ ऑटोजोम्स हैं ड्रोसोफिला में, ठीक है. लेकिन फोर्थ क्रोमोजोम, listen again, fourth chromosomes being the dot chromosome, that is mainly not considered for determining sex of the fly, okay. And therefore most of the time you will hear that in male drosophila there are 2 sets of autosomes and 1 X chromosome, and therefore the ratio is X divided by 2N is going to be 0.5. ठीक है, किसी को यहाँ पे क्वेश्चन है? देखो अब धीरे धीरे जैसे जैसे more techniques are evolving, more understanding is coming into the picture, now we know that even this dot chromosome, which is referred as fourth chromosome, has many important genes. Okay, now we know. But earlier it was thought that two sets of autosomes, they are playing important role. Okay, so we will consider this observation only, and therefore the sex is determined by number of X chromosome divided by 2 n. Okay, so in male the ratio is going to be 0.5, and in female it is going to be 1. Okay, and if the sex index is 1, then I'm sorry, uh, is intermediate, for example, let us consider 0.67. Okay, between 1 and 0.5, then this particular individual is going to be intersex. Additionally, when the ratio is going to be more than 1, that is 1.5, the sex is going to be, can you please tell me what it should be?
Super
Female.
super
Meta.
female. Female.
Right, and it is also known as super female. So both the words can be used, super female or meta female. But if the ratio is below 0.5, the sex is going to be super male or meta male, and they have serious developmental effects. And many, when there is a meta female or meta male, then they even do not come out of the pupal case. So there are significant developmental defects have been observed if the ratio is being altered. Now, you can also try multiple ways. What is going to be the multiple way? You can try yourself what will be the sex of the fly when there are 3x chromosomes and 4 sets of autosomes. Okay, and then what is going to be the sexual phenotype? Another one, if there is XY and at the same time there are 3 sets of autosomes. So it is, you have to play with different karyotypes of drosophila as well. Okay, because in exams it's a very common question which can be asked. So no one is going to ask you कि male or female का ratio कितना होता है. They might give you some karyotypes, and then you have to tell about the final phenotype of the fly. Okay, so as I said, uh, if there is any imbalance in the ratio, a very good example have been cited in many places, like the genotype is XY, but autosome number has been increased, and the ratio is going to be completely different. Now it comes to 0.33. And that is where you can imagine multiple types of experiments were performed to clearly show that it is not only the ratio of the X chromosome, it is the ratio between X chromosome and autosome which is going to determine the sex of the fly, irrespective of presence or absence of the Y chromosome. Okay, so the flies which have only single X chromosome and 2 sets of autosome, they are going to develop as male. That was one of the biggest understanding. Okay, so these males, now please listen to me carefully. They had 1 X chromosome and normal sets of autosomes. So ratio is 0.5, perfect. So phenotypically they are going to be male, but these males are sterile. There comes the maleness and the male functioning. Developmental biology clearly says, very clearly, and even genetics says very clearly, that whatever the chromosomal constitution be, if male is a male, it has to perform its function. That is, it has to produce the progeny, it has to have the capability to mate with the female and produce next generation. But look at the consequence. If there is single X chromosome, the male is going to be male phenotypically, but in terms of performance, it's not going to function like a male. Okay, and this observation further gave depth to the understanding that Y chromosome is not going to determine sex in the drosophila, okay, but Y chromosome is important for its mating behavior or fertility. Any question? ये आप लोगों ने पढ़ा है पहले?
Yes, ma'am.
Yes,
Yes,
ये सब
ma'am.
ये
Yes,
सब
ma'am.
पढ़ा हुआ है ना? हाँ?
Yes, ma'am.
So,
Yes, ma'am.
अच्छा, तो अब ये बताओ कि कौन सा जीन है which is very well studied and in case of drosophila and that is going to have influence on sexual phenotype. Can you please tell me?
Ma'am, SLX is XL gene. SXL.
Okay. Anyone else?
Ma'am,
Sex
transformer
lethal.
gene.
Yeah. Okay. So very nice. So there is a gene which is known as transformer. There is a role of XXL as well, and we will talk about that. But it is very, very important to know that transformer is the gene, and if there is mutation in this gene, then it is going to convert a female, which is going to have totally normal ratio of 1 into phenotypic male. Okay, whereas there is another one which is known as double sex mutation, and this double sex mutation has the capability to transform normal male and female into flies with intersex phenotype. Okay, so double sex mutation is has the capability to transform both male and female but into intersex. The word itself is telling us. Okay, and transformer, it is going to convert female into a phenotypic male. So हमेशा आप लोग ये word सुनोगे phenotypically, but functionally is very very important. Okay, and there comes the proper ratio. Now, if you, so this is just a brief background about the fruit fly sex determination. When you talk about the sex determination in human being, I'm not going to go into it. All of you know that the presence of genes on the Y chromosome is going to determine the maleness. Okay, but I am not going to agree with this particular statement. Okay, why? Because last time when we were talking, we were discussing about SRY, है ना? SRY क्या करता है? हमने आपको बताया कि अगर a piece of Y chromosome is getting attached to X chromosome, now please switch yourself. Come to mammalian system. तो SRY जीन है, वो कहीं भी अगर X chromosome पे attach हो गया, it is going to give a male phenotype. Okay, but then later on there was serious understanding that it is not only SRY gene which does the job. Okay, so we will come to that part very soon. Let us understand little bit about the Y chromosome in historical terms. So if you talk about human Y chromosome, early in 1900, the human females were thought to have uh, uh, extra chromosome. No one knew कि क्या है, किसको, सिर्फ based on the appearance of the chromosome, they were saying it is X chromosome, it is Y chromosome. So number was not known. So people were saying कि females के पास कुछ ज्यादा chromosome है. कैसे? उन्होंने कहा कि if male has 46, then females are going to have 47. I'm talking about early 1900. But then in 1920, another scientist whose name was Theophilus Painter, he observed that there was a small Y chromosome. But total number, because of the lack of genetic techniques, he still said it was between 45 to 48, but he was not clear about the exact number at all. Okay. In 1956, human chromosome number was set at 46 by Geo and Levan. And this happened only because there were improved staining techniques, bending techniques, and finally it was concluded that there are 22 pairs of autosomes and one pair of chromosomes. Okay? So you can imagine, even the number was not known because of the limitations of the techniques. But still, there was a question that whether Y is going to determine the maleness or absence of two X chromosome or presence of one chromosome is going to determine the sex. Okay? And finally, with various observations, it turned out that no, it has to be two X chromosome from female for a female phenotype and for function as well. Only one X chromosome is not going to work despite of absence of Y chromosome because these females were sterile. Okay. Another observation was there. One X chromosome is equally important for development. If there is going to be only Y chromosome, then fetus is going to be aborted. Okay. So for developmental purpose, what is the basic requirement? Basic requirement is to have one X chromosome at least. Okay. And if one X chromosome is not there, then despite of presence of Y chromosome, the fetus is not going to develop. Right? अब अगर हम एक X क्रोमोसोम की बात करें तो कौन सा सिंड्रोम है जहां पे there is only one X chromosome which is present?
Turner
syndrome.
So that if there is only one X chromosome, then development is possible, but that is going to lead to several problems which are actually related with the sex. So determination is one thing, functioning is another thing, right? And what we care the most, we care that determination should be proper, but at the same time, the female has to perform function of the female and male has to do the function of male. So what happens, just to brush up our memory quickly, in Turner syndrome, one of the thing which is observed in most of the syndrome is about the intelligence. Okay, it has been observed that in Turner syndrome the intelligence is almost normal. Okay, and this is considered as a syndrome which has to be looked into the details because prevalence is quite high. This is seen in 1 of 3,000 female birth, and now the number is increasing further. Okay, phenotypically they are female and they have rudimentary ovary as well, but uh, because of the Turner syndrome, and in another word, because of the lacking of two X chromosome, these females are sterile. And this karyotype of Turner syndrome was observed by, do you know about this part as well? Who observed Turner syndrome first time? Okay, so it was C.E. Ford in 1959, and he showed that Turner syndrome patients, they have only 45 chromosomes. And when he patiently observed the karyotype, he said that there is only single X chromosome. And this chromosome complement he referred as XO. Okay, and half of the individuals with Turner syndrome, they have monosomy, that is X chromosome, which means each cell in the individual's body is going to have only one copy of the X chromosome instead of the common one, which is the presence of two X chromosomes. है ना, ये सब चीजें तो पढ़ी हैं. We are just trying to brushing our memory. So, in addition to that, people started looking into the causative factor which is leading towards Turner syndrome. Now, think about another possibility of dosage compensation, right? Dosage compensation क्या होता है?
Inactivation of one of the
Forget about the
chromosomes.
definition part. No, it is not inactivation only. That is the process, that is the mechanism, that is part of dosage compensation. Basically, what happens, I'm not talking in a scientific words, just in a layman language to explain you. Dosage compensation. That means the genes which are present on two X chromosome of the female versus one X chromosome of the male has to be compensated. Okay, because if you consider the genes which are expressed on the X chromosomes of the female are going to have much higher expression as compared to the male, right? And therefore, one of the X chromosome has to undergo inactivation, which is, I said just now, is a process. Okay, now think about Turner syndrome. What must be happening in Turner syndrome? There is only one X chromosome. So dosage compensation must be happening there, or no?
No, ma'am.
No? Okay. And how about polyx chromosome? Polyx syndrome है ना? There are multiple X chromosomes. There can be 4, there can be 5. And then what happens? What is the default state? Dosage compensation?
Ma'am, in that case, only one X is active and other are inactivated.
Other ones are inactivated. है ना? ऐसा होता है ना कि other ones are inactivated. Now, how the cell is going to know that I have to inactivate only one? How it counts? कैसे पता कि यहाँ पे 4 हैं, यहाँ पे 5 हैं? कैसे पता चलता होगा? क्योंकि नॉर्मल जो फिनोमेना है, that is presence of 2X chromosome, and now it is totally abnormal condition that there are 4X chromosomes, there are 5X chromosomes, and if you talk about Klinefelter syndrome also, there are more than 1X chromosomes. So how cell would decide?
Ma'am,
Huh.
ma'am, I think there is a mechanism or there is a certain marker gene present in the X chromosome specifically, which basically dictates if the chromosome needs to get activated or inactivated. So according to that, the number of chromosomes which are to be inactivated is controlled. I think the X is gene, if I'm not wrong.
If you are saying that we are discussing right now, you said there is a marker gene which must be present on the X chromosome, and this marker gene must be deciding about the inactivation. Am I right? I understood you right?
Yes, ma'am. Yes, ma'am.
So ये marker gene तो सभी में है. आपके पास अगर 5 X क्रोमोसोम हैं तो पांचों में मार्कर जीन है. तो ये मार्कर जीन कैसे डिसाइड करता है कि एक को हमें इनएक्टिवेट करना है
Ma'am,
या
there
I'm
are
sorry, एक को एक्टिव रखना है, बाकी सबको इनएक्टिवेट करना है?
ma'am, there are
X inactivation centers that are present on the long arm of X chromosomes.
तो वो भी सेम थिंग इज ट्रू देयर आल्सो. भाई मेरा क्वेश्चन ये है कि एक ही एक्टिव कैसे रहता है? कुछ तो होता होगा ना. एक्सआईसी तो दोनों में है ना.
Ma'am,
हाँ.
there is a hypothesis known as Leon's hypothesis. I guess ये random inactivation वाला होता है. That parental और maternal में से paternal और maternal में से किसी भी एक X chromosome का inactivation हो जाता है during the cytodifferentiation of the embryonic tissue. तब वो
Okay.
random होता है ये चीज.
हाँ. तो now I am going to put another understanding for you, and it is my habit that I put the seed beforehand, okay, so that you will start thinking about it. Now basically what happens, ये सब चीजें जो भी आप बता रहे हो कि yes indeed all these things happen, except what the first student said कि there is a marker gene, okay, बाकी सब चीजें होती हैं, लेकिन एक ही कैसे एक्टिव रहता है, that is very very interesting. Basically there is a blocking factor, and this blocking factor is coming from autosomes. Okay, what is the job of this blocking factor? Blocking factor protect करके cover करके रखता है एक X chromosome को. ठीक है, we'll go into the details of this, and there are certain genes which are going to decide that okay, now blocking factor should be released. Interestingly, there is limited blocking factor, very limited, which is being released by the autosome, and this blocking factor is good enough only to protect one X chromosome. Only one X. कैसे ये कहा गया? एक बहुत सिंपल एक्सपेरिमेंट किया. उन्होंने क्या किया कि अगर ठीक है, अगर ब्लॉकिंग फैक्टर ऑटोसोम से आ रहा है, और हम लोग अनफॉर्चुनेटली अगेन आई एम सेइंग दैट वी विल नॉट बी एबल टू गो इनटू ईच स्टेटमेंट इन डिटेल, लेकिन आई वांट कि जितना ज्यादा से ज्यादा आप लोग नई चीजें समझ सको, दैट वुड बी नाइस. ओके, सो क्या किया गया कि इन बाय जेनेटिक टेक्निक्स, इंस्टेड ऑफ कितने पेयर्स होते हैं ऑटोसोम्स के ह्यूमन बीइंग में?
22 pairs.
22.
22. Now, just I'm waking you up. So 22 की जगह उन्होंने 44 कर दिया, डुप्लीकेशन कर दिया पूरा. And then it was very, very interesting to observe that instead of one X chromosome, two X chromosome remained active. Okay, so हमने आपको बहुत सिंपल तरीके से बताया, but if you will go through the paper, they are talking about the the techniques, the mechanisms, and all these things. But right now, with Turner syndrome, you have to understand one thing, that their dosage compensation is involved in Turner syndrome. Another concept which I gave you is the one of the X chromosome remains active due to the blocking factor which is released by autosome. And this blocking factor, the word itself is saying, is going to completely protect the one of the X chromosome only. Okay, so yes, we will, and there are some genes which are required for activating these blocking factor and so on and so forth. So if you talk about Turner syndrome, then there are no known cases in which a person is completely missing both the X chromosomes. And these syndromes give us a lot of idea about sex chromosomes, their functioning, and so on and so forth. And from there, that no patient with Turner syndrome was without a single X chromosome. So then it was understood very clearly that embryos missing both the X chromosomes, they are spontaneously aborted during early stages of development. Okay. Researchers still could not determine which genes on the X chromosome which is associated most of the features of the Turner syndrome. Working, people are working, but they have identified one gene which is called SHOX, SHOX. And this SHOX is important for bone development and growth also. It is observed that loss of one copy of SHOX is the causative factor of short stature and skeletal abnormalities which were observed in women with Turner syndrome. Okay, because आप लोगों ने पढ़ा होगा कि they are very thin. The females with Turner syndrome, they are very thin. So in addition to that, inheritance pattern was also observed in details in Turner syndrome, because most of the cases of Turner syndromes, they are not being inherited. Okay, whenever this condition, that is Turner syndrome, is resulting from monosomy of X chromosome, the chromosomal abnormalities are going to occur as a random event during the formation of reproductive cells, okay, in the affected person's parent. So an error in cell division, which is again called as non-disjunction, it can result in reproductive cells with abnormal number of chromosomes. And a simple example, if you would like me to give the example, if there is an egg or sperm cell and it can lose a sex chromosome as a result of non-disjunction, then these atypical reproductive cells are going to contribute to the genetic makeup of the child, and the child is going to have only single X chromosome in each cell and will be missing the other sex chromosome. And this whatever I am saying, it is going to be clear to you when we talk about non-disjunction. आप लोगों ने ये सब पढ़ा है, लेकिन कई बार जब कॉन्टेक्स्ट में उसकी बात की जाती है तो फिर से you try to remember what you have studied before. Now, if you talk about mosaic Turner syndrome, that is also not being inherited. In affected individual, this mosaic Turner syndrome, it occurs as a random event during cell division, and this happens only in early fetal development. And as a result, some of the affected person's cells, they are going to have the usual 2X chromosome, okay? And other cells are going to have only one copy of X chromosome. So I think you must have heard about mosaic Turner syndrome as well, because some of the cells are going to have two sex chromosomes, but some of the cells are going to have only one copy of the X chromosome. Okay. So there are multiple other sex chromosome abnormalities which are also observed in females with X chromosome mosaicism. However, the Turner syndrome is also known to be caused by partial deletion of the X chromosome. And whenever there is partial deletion of the X chromosome, that means there are going to be two X chromosomes, but one of the X chromosome is partially deleted. And in this case, it is considered as inherited, because now there won't be any situation of non-disjunction. Only part of the X chromosome is going to be deleted, and therefore it is going to be passed from one generation to the next generation. Okay, anything else related to this? So I think you need to go for another class. We will talk little bit about some of the genes which are involved in Turner syndrome tomorrow. Okay, this